Next up is Oral 25, which is Valley Center, Kansas. This is a Mission 3B flight experiment, which also came down in March.
The title of this experiment is The Effect of Microgravity on Bacterial Growth and Its Resistance to Antibiotics. My name is Cole Klinkhammer, and these are my co-principal investigators, Wesley Crow, Logan Burks, and Samuel Sheehan. We are from Valley Center, Kansas. Next, please. The idea of the experiment was to not only test a specific bacteria's growth in space, but also to test its resistance to antibiotics. While working through the details of this experiment, the question was raised, will the bacteria be less susceptible to antibiotics after it has grown under the influences of microgravity? Now, in previous experiments using E. coli, the bacteria had a cell population of 88% more than the control. Also, in our pre-flight tests, we found that the bacteria were already growing resistance colonies while being grown under the effects of gravity. Now, taking all of these known results, we hypothesized that under the growth of microgravity, the bacteria would become even less susceptible to antibiotics. Next, please.
The bacteria we used in our experiment was Staphylococcus epidermidis. There were two main reasons why we chose this bacteria. One reason was it was a facultative anaerobe, which means it can live with or without oxygen. It is a normal part of the human skin flora, and therefore it's harmless. The second reason was that we were able to receive the bacteria freeze-dried, which allowed us to control the bacteria when it would be activated in the International Space Station. Before it could go up into space, we had to do multiple steps of pre-flight stages. Well, first, we sterilized our FME and all of our tools. Next, we crushed bacteria and loaded it into one end of the FME. Next, we took a sterile syringe to load our broth into the FME and then clamped the broth in shut. We prepared two FMEs, one for spaceflight and one for ground testing. We had the astronaut shake the FME 3 times for 20 seconds to allow the broth to completely mix with the bacteria. Once the FME returned from space, we made several observations and noticed the differences between the FME from our spaceflight and our ground FME. In order to study this bacteria and its reaction to the antibiotics, we created culture plates by pouring agar into petri dishes. We then swabbed the bacteria grown both in space and onto Earth, onto culture plates. On the culture plates, we used 3 types of antibiotics: penicillin, erythromycin, and vancomycin, which were small discs. Each of the 12 culture plates were then split into quarters. We placed one of each antibiotic disc into its own quadrant. The 4th disc was a control disc. The culture plates then sat in the incubator for 48 hours at 37 degrees Celsius, and then we analyzed our results.
Next, please.
Before putting the bacteria into the culture plates, some of the observations that were made were the FME, which contained the bacteria from the ISS, was darker in color, had a fouler smell, and was more gelatinous in form, while the FME that contained the bacteria grown on Earth was in more of a liquid state, had a weak smell, and was pale in color. When we observed the bacteria and their zones of inhibition, it was noted that the bacteria grown on the ISS was more virulent or potent and had smaller zones of inhibition around the antibiotic discs. The resistance colonies in the culture plate that contained the bacteria that was grown on the International Space Station were more dense than the resistance colonies found in the culture plate with the bacteria grown on Earth.
With every part of the experiment remaining the same, except for the independent variable, gravity, we have come to the conclusion that bacteria grows better in microgravity based on our results shown. One supporting piece of evidence in this difference between is the bacteria grown on Earth and the bacteria grown during our pre-flight tests. During the pre-flight experiments, the bacteria wasn't allowed to grow as long, and so there were thicker growths on the petri dish. During the actual ground test, the bacteria had a longer time to grow, resulting in thinner groupings on the petri dish. Even though this happened to the ground test, though, the space test yielded different results. The bacteria actually seemed to thrive during its extra growing time on the ISS, resulting in much thicker colonies on the petri dish. We believe that this phenomenon can primarily be credited to microgravity. Both bacteria were activated at the same time and had the same amount of time to grow. thus showing that the bacteria grown in space were still living and appeared dense and strong. As stated previously, the bacteria on the ISS showed smaller zones of inhibition, an average of 0.7 centimeters smaller to be exact. Next slide, please. While they may seem really small, while this difference may seem very small, it's actually quite considerably large in difference compared when it comes to microbiology. According to the various results we observed, it was easy to decide that bacteria grew better and stronger while under the influences of microgravity. Next, please. As shown throughout this presentation, through numerous examples, we have exemplified that bacteria grows better under the influence of microgravity, making it more resistant to antibiotics. But why? Our hypothesis is that the lack of gravity allows for the bacteria not to settle, allowing it to access all of the nutrients and grow more freely. If we had the opportunity to do this again, we would try using different bacteria or possibly viruses. We feel that based on our results, this may affect safety precautions taken upon astronauts when they return from space, and also the bacteria or the antibiotics. It might affect the strength of the antibiotics sent back into space when people go to space. Next, please.
Over the course of this experiment, there are many different learning opportunities, from microbiology to responsibility. I can speak for all of the group by saying that we have benefited greatly from this opportunity. Of course, it wouldn't be possible without the help of our sponsors, who we would like to thank now. This concludes our presentation.
Questions for the Valley Center, Kansas team? Okay, let's thank Valley Center, Kansas.